Hello, welcome to BioLab. This is going to be a brief lecture on safety and scientific measurement so that we can get right to work when we get to lab. Here's our objectives for this week. You can review this additionally on Moodle. But we're going to be doing some activities about safety and the metric system, using equipment, and orienting yourself to the lab room. Okay, this is from your lab manual. Table 1. Here in the United States, we tend to use American customary units, so inches, miles, gallons, and we do temperature in Fahrenheit. The rest of the world, and in Science Lab, uses the metric system because it's so much easier to do conversions between units and make comparisons and do the calculations of it. And I think that you will appreciate this once you get used to it. So don't freak out if you're not familiar with it. I'll walk you through it, and it's really not that scary, I promise. In your lab manual, this is also Table 2, metric prefixes, unit decimal values. Within the metric system, we can easily convert between units because it's base 10. That doesn't work in American customary units. This chart looks really huge and scary. Don't freak out about it. This is the next figure in your book. This breaks it down. This is more of what you'll need to be using immediately in biology lab and other science classes. You may use other versions, but these prefixes are the ones that I would like you to be familiar with, from kilo down to micro. We'll do some practicing together in class, but the general idea is that starting at the basic unit, if you want to convert to a smaller unit, you are basically moving the decimal point to the right or multiplying the number by whatever number you've got here. If you're more math-minded and want to do the math, please go for it. Otherwise, I'll walk you through moving decimal points, which is doing that. If we want to go from a smaller unit to a larger unit, we move the decimal point to the left or divide by that number. So if we had 2 meters and we wanted to know how many kilometers that was, we would divide 2 by 1,000 or move the decimal place over 1, 2, 3 units to get 0.002 kilometers. Again, we'll do that in class because some people kind of freak out about that. I like to have mnemonics whenever I can. Here's one that might work for you for memorizing the kilo through milli. And you just need to know that micro is another 3 to the right. It's got some abbreviations there for you. The other thing I want to interject here is that as we're doing conversions, if this isn't making sense to you and you want more help, the CAPS tutoring is available, but they have a whole math tutoring division, and they can really help you. They'll do a much better job of explaining the math behind this than I could, and I don't want to take a whole lot of time going into it. But if you're struggling with that, please go make an appointment there. One of the tools we'll be using today to measure temperature, which will be always in Celsius in biology lab, is a LabQuest 2. There's a video here. If you would like to watch it and learn how to use it ahead of time, go for it. It's very simple. And again, I'll talk you through it, but I wanted to point out that video is available ahead of time if you're one of those people who likes to watch videos. And this is a metric ruler, not to scale obviously. but you'd be amazed at how many people don't know what these things are. Okay, so the numbers on the top here are in the longer lines are the centimeter marks, and the little lines in between them are millimeter marks. This will help you if you didn't already know that. It's not a problem if you didn't. The other thing we'll be going over is some basic graphing skills, and then talking about correlations between data. This is a scatter plot of human height versus weight. I don't think you can see the height at the bottom, sorry, but it's there. And so each individual was plotted based on how tall they were and how much they weighed. Isn't that interesting that the weight is also supposedly in centimeters? I don't like this graph. I'm going to have to find a different one. We're going to make one in class. It'll be much, much better. But you want to start thinking about how the data points are distributed on a graph. Are they? They've drawn a line of best fit here. How close are all the data points? Is it all right on a line or not? That how it fits the line is called the correlation, the relationship between two sets of data. And we're going to be asking, is the data have a positive or negative correlation? And then is it a strong, weak, or moderate? Or no correlation for that matter. Again, we'll go over our data and look at some more of this in class. If you're taking statistics, and as you go on in biology, you will need to use statistics to further describe the correlation. And R squared is the terminology that is used there. If you're not taking statistics, don't worry about it right now. But I do recommend that you learn about that at some point before you advance too far in biology, certainly. Helps us determine if our data is agreeing with our hypotheses. And there is the results from a couple years ago comparing foot length and height of HCC students and looking for a correlation. So we'll examine that more in class. Anyways, I hope that this was helpful. We'll switch back to the webcam. Hey, hi. So there's hopefully you were also able to do the practice quiz and off the syllabus and the safety video. And you're going to want to have a copy of the list of things that you came up with so that we can compare notes in class. And then I'll grade them. I'll grade the things that have been submitted later. But, take care, and I'll see you in class.